Have you ever stopped to wonder how bacteria are just so incredibly tough? I mean, they're some of the most resilient organisms on the entire planet. It's not just that they're small or reproduce fast. No, their secret is a highly sophisticated, microscopic suit of armor. Today, we're going to decode the secrets of that armor, the bacterial cell wall. Seriously, think about it. From boiling hot volcanic vents to the deep freeze of the Arctic, bacteria are absolutely everywhere. So what gives these tiny little organisms such an edge? The answer really lies in their protective layers, especially a structure that acts as both a rigid shield and a flexible cage. That's the cell wall. It is the absolute key to their survival, and we're about to see exactly how it works.
But, to really get a handle on the armor, we first need to understand the body it's protecting. So, let's start with a really quick tour of a bacterial cell before we zoom in on the main event, its primary defense system.
Okay, so here's a peek inside your typical bacterium. You can see all the basics, the DNA just kind of coiled up in the middle, ribosomes floating around, but our focus is right here on the edge. You've got the inner plasma membrane, and then, wrapped around that, the star of our show, the thick, absolutely essential cell wall.
So, what's the big deal with this wall? What does it actually do? Well, first off, it gives the bacterium its shape, kind of like an external skeleton. But more importantly, it stops the cell from literally exploding. See, bacteria often live in watery places, and without that wall, water would just rush in and boom, the cell would burst. It also acts as a bouncer, keeping out toxins. And for the pathogenic bacteria, the ones that make us sick, this wall is a critical part of their toolkit.
All right, let's get into the nitty-gritty. What is this incredible armor actually made of? Well, almost all bacterial cell walls are built from a fantastic material called Peptidoglycan. The best way to think of it is like biological chainmail, or maybe rebar-reinforced concrete. It's unbelievably strong, but also has to be flexible.
Peptidoglycan, sometimes called murin, sounds complicated, but it's really just a massive molecule made of smaller repeating units of sugars and amino acids. And when I say massive, I mean it. They all link together to form one single, enormous, bag-like mesh that wraps around the entire bacterium. It's an engineering marvel, really. It has to be tough enough to handle huge internal pressure, but also porous enough to let nutrients get in.
And this is basically how it's built. You start with the basic building block you see there on the left. Just a couple of sugars with a little tail of amino acids. Then you link those blocks together over and over and over again until you get the vast, strong, three-dimensional mesh you see on the right.
Now, this is where it gets really clever. This is just a brilliant piece of molecular defense. See, most life on Earth, including you and me, we use what are called L amino acids. Our bodies have enzymes specifically designed to recognize and break them down. But bacteria pull a fast one. They build their cell walls using D amino acids, the mirror image. It's like trying to open a lock with the wrong key. Most enzymes just can't grab onto them, which makes the wall incredibly tough to break down.
Okay, so you have all these strong chains. How do you hold them all together to make that mesh? Well, there are two main strategies. Some bacteria go for a direct bond, linking one amino acid chain right to another. Others use an indirect approach, using a little connector, a short chain of extra amino acids called a peptide interbridge.
And you can really see the difference right here. On the left, that's your direct, rigid connection, simple and strong. But on the right, you've got that flexible little bridge holding the two chains together. It seems like a small detail, but this subtle difference in construction completely changes the wall's properties, and, as we'll see, it's a key distinction between different types of bacteria.
So let's see how these building blocks come together in the first of two major architectural styles. First up, the gram-positive cell wall. And I want you to think of this one as a fortress. It's thick, it's imposing, and it's built for pure, raw defensive power.
Whoa, just look at that. A gram-positive wall has a truly massive layer of peptidoglycan. We're talking up to 80 nanometers thick. But it's not just one big solid block. You see those molecules running through it? Those are called teichoic acids, and they act like rebar, reinforcing the whole structure and giving it even more integrity.
And these teichoic acids are real multitaskers. They act like anchors, locking the cell wall to the membrane below. They also give the whole surface of the bacterium a negative charge, which is important. And yeah, for the pathogenic types, these molecules can even help them latch onto our cells to kickstart an infection.
Okay, so from the fortress, we move to our second style, the gram-negative architecture. And if gram-positive is a fortress, then gram-negative is more like a medieval castle. It's all about layered defenses. It's got a thinner main wall, an outer wall, and even something like a moat.
Now this looks way more complex, right? And it is. The huge difference here is that second outer membrane. The peptidoglycan layer, the main wall, is actually pretty thin, and it's just sandwiched in this space between the inner and outer membranes. It is a totally different defensive philosophy.
And that space between the membranes, the castle's moat if you will, is a really big deal. It can make up 20 to 40% of the cell's entire volume. It's not empty space either. It is a bustling hub of activity, packed with enzymes and proteins needed to grab nutrients and build the cell.
Now, that outer membrane of gram-negative bacteria is famous for this one unique, really powerful molecule, lipopolysaccharide, or LPS. It's built in 3 parts. First, you've got lipid A, which is the anchor stuck in the membrane. Then there's the core polysaccharide, which links everything together. And finally, sticking way out into the environment, is the O side chain. This is also called the O antigen, and it's basically a flag that our immune system is trained to recognize.
And LPS is incredibly important to the bacterium. It helps stabilize that outer membrane and acts as a barrier, blocking a lot of antibiotics and other toxins from getting in. But it has a darker side. That lipid A part is a potent toxin for us. When gram-negative bacteria die and break apart, they release it, and that's what can trigger fever, inflammation, and even life-threatening shock during an infection.
So, as if all of that wasn't enough, some bacteria decide to add even more layers to their armor. You can think of these as optional extras, like adding a coat of camouflage or a slick of oil to the main suit of armor.
This table breaks down the main types really nicely. A capsule is a neat, well-organized layer that helps pathogens basically hide from our immune cells. A slime layer is way more diffuse and disorganized, and it's great for helping bacteria stick to surfaces. And then you have the S layer, which is this amazing crystalline layer of protein, like microscopic floor tiles, that gives rigid protection against all kinds of environmental stress.
So when you really step back and look at the sheer complexity of it all, from the clever molecular trickery of D-amino acids to the fortress-like walls of gram-positives and the complex, layered defenses of gram-negatives, you're looking at an absolute masterpiece of evolutionary engineering. This armor has been battle-tested and refined over billions of years, which leaves us with a pretty critical question for modern medicine and science. How can we possibly outsmart it?